Hi students, welcome to MyRank Tuitions Physiology. We are talking about gastrointestinal physiology. I'm Dr. Ashwin. We learned about the various gastrointestinal secretions, the salivary, the gastric, the pancreatic, intestinal. We also learned about the secretion of the bile from the liver. But the point is, we must understand the structure and functions of the liver and gallbladder in certain detail so that we will be able to know how intricate and how complex are the anatomical, histological, and the biochemical reactions which are happening in the liver. And liver happens to be one of the most important organs in our body, especially concerned with digestion and absorption. Okay. Is it only with digestion and absorption or does it do anything else? We will learn it in this class. Coming to exam point of view, liver and gallbladder is usually an essay question from the GIT. Okay. So the question will directly come like a descriptive question. Briefly describe about the hepatic lobule and the formation of the bile, functions of the bile or functions of the liver and so on, and add a note on jaundice. Or the question can come as a clinical scenario wherein it will be asked a middle-aged man has come to hospital with yellowish discoloration in the sclera and the skin, and then the doctor suspected jaundice and the laboratory report is like this: the direct bilirubin so and so, indirect is so and so. Now it will be asked to you comment on the condition of the person and give your differential diagnosis. Okay. The term that will be used is differential diagnosis. That means you will be able to tell what all possible conditions can lead to that. So what we will do in today's lecture is that we will spend a significant amount of time in the last sections of our class where I will be telling you the differential diagnosis of jaundice. Okay. That's an important thing for you, which you can make a note in your textbook then and there. And the others will be certain descriptive points what we are going to learn. We will start today's class by knowing the functional anatomy of liver. Liver is a wonderful organ. I say it as wonderful organ because if you damage the liver to an extent, it has the capability of regeneration. It is said that the right and the left lobes of liver what we have, probably by this time you must have dissected the liver and carried it in your hands. Okay. So it has a bigger right lobe and a left lobe, a smaller left lobe. The point in the regeneration, what they say is 2/3 of the liver is actually extra, which is there with us. It is enough that you can survive with 1/3 of the liver. This reserve principle is not new to liver. It is there everywhere in the body. Even one kidney is sufficient for us. One lung is sufficient for us. The others are only just for reserve. Okay. So now coming to the liver, if the liver is damaged or insulted, as I told, it has a capability for regeneration. So it will grow on its own. But there is a condition. The liver has actually grown into a lattice or a network, okay, which is already created, and the cells were nicely going into it and then they got arranged. If that network or lattice or an extracellular matrix support is gone, then liver cells go mad. They don't know where to grow and where to go. Okay. So that preservation of the matrix is important. Only then the liver can regenerate. What is that matrix? Why did the cells have to go into such a intricate pathways? I will be trying to tell to you in this class. So the bile is the one which is a very important product that the hepatocytes will synthesize and then produce. Okay, bile is the product. Whereas the hepatocytes will do all other functions. They keep getting the portal blood and then they do carbohydrate, fat and protein metabolism. They keep synthesizing proteins. That's there. But bile is something very special to them. Why? Because they are synthesizing bile into a separate channel. That is interesting. You have to understand that. Like for example, say it is a hepatocyte. Now my hands I am putting it, that's a hepatocyte. It will receive the portal blood, it will receive hepatic artery blood so that it gives oxygen and nutrients, it will get the digested absorbed nutrients. So what this liver cell will do then? So for example, it received glucose. It will store glucose in the form of glycogen. For example, it raises amino acids, it synthesizes proteins. Fine. It synthesizes these things and puts them into the circulation that goes into the general circulation, that is towards the inferior vena cava and into the heart, because ultimately heart is the one which needs to receive it. But the same cell is so clever that it has segregated its work. Say 70% I will do this work, but 30% I will do my work. That is what a hepatocyte thinks. So in that 30%, it does another synthetic mechanism called bile it produces and puts the bile into a separate channel, not into the previous channel. This is important. Okay, so that it is sending bile into a separate channel, it is sending the food material or toxins whatever into a separate channel. Such is the intelligence of a hepatocyte. And for this difference or the division of labor, maybe I can say, in each hepatocyte, the anatomy of the liver is also complicated so much. Okay, so the bile is secreted into hepatic ducts finally, which join to form a common hepatic duct, and then it is stored in the gallbladder for some time, and via a common bile duct it enters into duodenum. The opening is blocked or controlled by a sphincter of Oddi. And hepatic artery veins and the portal veins are all the things which are coming and meeting at the liver. How it is happening, let us see. This picture is showing you a very tiny part of the lobule of the liver. You will shortly see what's a lobule. Okay, so here what do you see is that a central vein is there in the center. Okay, it is a vein, so that means its duty is ultimately to collect the blood and take it to heart, and it's not going to come back. Okay, so into the central vein, what are the channels that lead into the central vein? This blue color things, like rivers go and meet into a sea. So this tiny rivers called sinusoids are going and meeting the central vein. Okay, so by the sides of the river, on the banks of the river, I can say the hepatocytes are situated nicely. So what these hepatocytes will do, if whatever the things they want to send it to the general circulation, they pour it into the sinusoid nearer to the central vein. So what is a general thing what they want to do? Just now we discussed, they wanted to synthesize a clotting factor, say factor 8, and they synthesize this factor 8 and put it into this, and that will go into central vein, goes to heart and gets circulated to the entire body. Okay, so this hepatocytes, but if they want to synthesize some glycogen or this, they want to synthesize glucose, they from the breakdown of glycogen or gluconeogenesis. So where is the store then? From where will they get it? They have to get it from the intestine, right? So from the intestine, the portal vein which has collected these products of digestion, it will send these products into this canalically, sorry, into this sinusoids on one end. So as you can see, the portal vein is also emptying its contents into this river called a sinusoid. So in the entrance of the river, these nutrients are all released so that they will be taken up by the hepatocytes as and when they require. And they keep sharing the substances amongst themselves and they synthesize new things and they keep pouring it into it. That's very nice. And these hepatocytes also require basically some oxygen for their survival. So hepatic artery is there which has come to the liver, which will also split up into its capillaries and merge its capillaries into the sinusoids. So the capillaries of the portal vein and the hepatic artery are merged to form a sinusoid. Okay. So through this, the oxygen will keep going to all the individual manufacturing units. Okay. Fine. So now you understand the portal vein and the hepatic artery. Once they come into the liver, they finally divide into branches, small branches, tiny branches, and ultimately they merge or fuse into a sinusoid or a bigger capillary like thing. Okay. So now what is the next thing I told? Of course, from the digested material, the hepatocytes will take it up, they synthesize and they push it into this sinusoid river that goes into central vein and finally into the heart. But they want to show there some unique mark, something like, this is my ability. So they show it in by means of synthesizing a substance called bile. So, which we have seen in detail in the last class during GI secretions. I told you bile has bile acids and bile salts. Okay. So, anyways, so these hepatocytes are synthesizing that bile. Where are they going to send it? If you can see carefully, the hepatocytes, between each hepatocyte, there are tiny, tiny holes, okay, tiny gaps. These tiny gaps are called bile canaliculi. Okay, and they keep pouring that products into bile canaliculi, and these bile canaliculi over the entire lobule, they have their own nice networking thing and they keep uniting with each other, and each other tinier things become bigger and bigger, and finally they open into a duct called a bile duct. And like that, there are many numerous bile ducts which will ultimately form into a bigger bile duct, and that goes through a hepatic duct into a gallbladder. Okay. So as you can see, a typical hepatic lobule has a central vein, that means an output zone, and a triad of bile duct and this, which is kind of input zone on one side. Okay. But truly speaking, the bile duct is also an output from this, but it is not into the general circulation. No, it's back into the intestines, right? So this is what you need to understand. We are not going to stop here. We will see the anatomy of the liver in further detail in the next two slides. Before that, let me tell you what is the uniqueness in the portal circulation. This you already know. Say for example, in the heart, from the abdomen, from the arch of aorta, see this is the arch of aorta, there is a renal vein, which we saw in the previous class. The vein, the renal artery, sorry, it will go, it forms afferent arteriole, a capillary, efferent arteriole, capillaries again, a venule, and comes back and joins the inferior vena cava. This is a kind of circuit, okay? Say in skeletal muscle, so artery to the biceps will go and vein from biceps will go. Simply, it's a usual circuit. But when it comes to a portal circulation, what you are seeing is from the heart, if you see from the aorta, there are three main branches, the celiac, the superior mesenteric and the inferior mesenteric, supplying different segments of the gastrointestinal tract. Okay. The liver, the spleen and the stomach, the pancreas, small intestine, colon, etc. So whatever these structures are producing, okay, the spleen, the stomach, the pancreas, intestine, colon, that means the products of digestion and absorption, they through the portal vein ultimately go to the liver. Okay. So hepatic artery will bring the good amount of oxygenated blood, and portal vein will get the nutrient rich blood to the liver. Okay. So the liver will then utilize them both, have its own products, and sends its products through the vena cava into the heart. Okay. Or it has a special product called bile, which it will send back into the intestine. Okay. Into the intestine. Right. The microanatomy of liver. So this huge liver is shown here. Now let's observe a small tiny bit of it and then magnify it. What is a characteristic of liver is that the tissue of the liver is arranged in benzene rings like hexagonal units called lobules, the liver lobules. Okay, they are hexagonal, as you can see, 1, 2, 3, 4, 5, 6. In the center of this lobule, there is a tiny vein which is going called central vein. Okay. So the lobule is all having the hepatocytes now. They're all hepatocytes, but not just like this dots, but they're arranged in a beautiful architecture, which I will show you. So the center of a lobule is having a central vein. The central veins of all the lobules will likewise, they arise, they emerge and they fuse and they enter into hepatic vein. Okay, and these hepatic veins are the ones which empty the blood finally into the inferior vena cava. Okay,